So thank you again for the invitation to give you an update on the ARM vaccine program we're developing. So this is something really exciting for us because it's really revamping, if I may say, the field of cancer vaccination using a cell that has never been thought to be able to basically make its way into the field of cancer vaccination, of course, using the technology that was developed by Defense Therapeutics, which has been really revolutionizing the world of molecular and modern medicine with new developments and new properties. So again, just a quick reminder of what Defense is really focused on. What was the initial idea of using the Acume platform? We all know that if we have any kind of biomedicine we would like to develop or use to treat a specific disease, these molecules, which could be protein, DNA, RNA, antibodies, will be eventually taken up by cells in a process called endocytosis, which is shown here. So this is really kind of a capturing of product from the extracellular environment into structures that are called endosomes, which will end up really degrading the captured molecule into smaller pieces, and these pieces will no longer be active in actually achieving the objectives they were initially designed for. So if you can imagine, for example, someone delivering an RNA-based vaccine, well, this vaccine could be ending in the endosome, being degraded and not being able to be released into the cytosol, where it could be translated into a protein and induce its effect. Same thing applies to antibody drug conjugates. Same thing applies to, for example, RNAi or proteins in general. So what Defense did is really come up with this smart and neat technology, which is basically a lipopeptide that can induce a chemical reaction to break the endosomal membrane, allowing these products to be released directly into the cytosol and then reach whatever organelle they're meant to. So this is, again, a key experiment, key slide, key data that really shows what happens, for example, if you deliver an antigen that is fluorescent to the cell. You can see that the cell is entirely green. We're not able to, this is delivering an antigen to a cell that is expressing a protein that is fluorescent because it's binding to carbohydrates. So carbohydrates are always found on the cell surface or specifically in the endosome or Golgi apparatus. So if you look at the cell entirely, you'll see that it's all green in a uniform manner. So there's no really depiction of where the product is in the cell. Now, if you pulse that cell with an antigen that has the Acume is going to be able to break those endosomal membranes, allowing the fluorescent protein, the galectin-3 GFP, to really move in and out of the endosome, and therefore now bind specifically to carbohydrates. And that's why you move from something that is entirely green into a cell that has, those different green puncti. So as you can imagine, this is a revolutionizing technology because it can basically be applied in vaccination for cancer and infectious diseases. It could also be applied in the context of antibody drug conjugates, but also, as I will show, could be used to come up with new technologies, new approaches, new unexpected properties that could bring us to another field that is away from the delivery of these products in cells. So, of course, I strongly encourage those that are interested to really read our key paper in Cell Reports Medicine, really demonstrating how this technology was basically used to re-enhance, if I may say, the field of DC cancer vaccination. Having said that, this technology is not only, meant for one platform, one indication. It's really highly versatile, and I put a lot of emphasis on the word versatility because this is really what the molecule does. It could be used to generate or be bioconjugated to any antibody drug conjugates. For example, commercially available ADCs that have failed phase 1, 2, or 3 for dosing properties. And in fact, we have right now a partnership with a European company that has generated a very nice ADC that actually works pretty well in vitro, but the doses that you have to deliver in vivo are quite high. So we have great evidence right now that we can really diminish the concentration by 30 to 40 folds, which is amazing in terms of, therapeutic potency. We've shown that we can use it for the design of protein-based vaccines. We're working right now on a variety of mRNA vaccines. we've shown that this molecule on its own, without being conjugated to any byproduct, is by itself toxic to cancer cells, and therefore that's the premise of the AccuTox program, which had a FDA clearance to go to a phase 1. And of course, what is really impressive is the fact that we can also use it to develop what we call cell-based cancer vaccines. I've spoken about the DC-based vaccination, but now I'll be talking about something that is completely novel and innovative in the field. So this gives you a general idea about the pipeline we have in development. We've got the ARM vaccine, ARM-X vaccine, which is the second generation we'll be talking about today. But we also talked about the AccuTox molecule that has this anti-cancer property, which has been cleared for phase 1 trial. This is something we're going to be working on this year. We've developed, a protein-based vaccine for cervical cancer. That's another interesting product that we will foresee as highly innovative in cancer vaccination, because it's not only a vaccine that will be able to protect from cervical cancer or head and neck, but it's also very impressive in terms of its ability to control therapeutically pre-established tumors, something that Gardasil 9, for example, can do at the moment. And of course, we have our ADC program and the mRNA vaccination program that are actively ongoing in the company right now. So, what's the point behind the ARM vaccine? What's different with respect to what has been done in the field of cancer vaccination using dendritic cells, which, as we know, have been used in the 90s for development of vaccination strategies for melanoma and prostate cancer? Well, mesenchymal stromal cells are in general cells that have been known for their capacity to suppress the immune system or have this ability to migrate to ischemic tissues and repair those. So this is what MSCs are known for, and there are more than 600, 500 studies really showing you how these cells are potent in regenerative medicine or immune suppression in general for multiple sclerosis, rheumatoid arthritis, GVHD, and whatnot. However, you have to keep in mind that these cells have characteristics that make them way more interesting than just using them in regenerative medicine and immune suppression. So they're easily harvested. Initially, we spoke about the bone marrow, but today we know we can isolate them from a variety of different tissues. They've got a very impressive yield of expansion potential. So with one single vial of 1 million or 2 million cells, you can end up having billions of cells in a matter of 5-6 days. Their safety profile is really fantastic, especially for the clinical trials or clinical studies that were conducted so far. They have this amazing plasticity. And that's why I was really interested in MSCs, because once you have a cell that has this kind of plasticity, it means you can actually drive it to go in one way or the other if you find a magic formula that can make them, to behave the way you want them. And that's why I think that they're a solution looking for a problem, because they could be adapted to your problem depending on what you're looking for. So we were actually the leading team so far demonstrating that we can reprogram MSCs either genetically or pharmacologically to make them behave as antigen-presenting cells. In other words, cells that could stimulate the immune system, drive a cytotoxic T lymphocyte response that can fight cancer or infectious diseases. And here are some examples we've developed so far. We have more studies that are actually cooking right now and will be published very soon. So what's really impressive about the AccuTox, if you recall the past presentations and what I mentioned earlier during the presentation, AccuTox is a derivative of the Acu molecule. So it's still a lipopeptide. It's been modified. But what's interesting about the AccuTox is that while we were investigating its mode of action as an injectable in animal models, I mean, we've demonstrated that it can kill cells through immunogenic cell death, which is a form of death attracting the attention of the immune system to mount an antigen-specific immune and inflammatory response. We were able to demonstrate that it produces ROS, reactive oxygen species, which affect really endosomal structures because it destroys those, and it destroys, these intracellular transportation mechanisms. And it's also involved in blocking repair mechanisms that are really acting on the DNA once you have this kind of a genotoxic effect. But while we were investigating all this, we also noticed through transcriptomic analysis that AccuTox enhances antigen presentation. And in fact, as we speak right now, we're conducting studies to demonstrate that if you treat cancer cells with low-dose Acitox were able to actually show, if they were transplanted in animals, that they grow much slower than their counterparts that have not been treated. So that's another evidence that this molecule really makes non-immunogenic cancer cells immunogenic and visible to the immune system. So that's if you're thinking about the molecule as a non-bioconjugated, freely available injectable compound. However, we said to ourselves, okay, that's interesting, but since we're working in antigen cross-presentation and cell-based vaccination, could we, for example, use that molecule to convert MSCs into antigen-presenting cells the same way we did in the past with the A1 molecule, which is another Acuum variant, which was basically inducing what we call the ARM vaccine. Now we're going to be talking about the second generation ARM vaccine we called ARM-X for the use of Acitox. So really the first thing we did for that study is, first of all, identify the dose that doesn't kill the cells, because we know Acitox is pretty harsh on cells. If you add a specific concentration, any cell will die. So we wanted to really identify the dose in vitro that doesn't kill the MSCs. And we noticed that, we can go up to 25 micromolar. After that, the cells start dying and you end up having nothing in your Petri dish. So once we identify these concentrations or doses of the Acitox, we then ask the question, could, for example, the Acitox enhance antigen capturing from the extracellular environment? So to answer that question, we use a fluorescent ovalbumin protein. So that's a protein that fluoresces once it's captured, or if it's like in a cell. And as you can see, if you look at a regular MSC that is not treated with the Acitox, you get a signal. So cells capture the fluorescent ova naturally. But then if you add the Acitox at different concentrations, you see that the histogram is shifting towards the right, meaning that the signal is there, but it's actually amplified, it's enhanced. And you can see that, the shift is getting stronger and stronger once you increase the concentration of, once you increase the concentration of the compound. Now, notice here that we have used basically 50 micromolar. In fact, although we see a very nice increase, I'd like to remind you that in this case, the cells are not feeling well, they're not really proliferating anymore. So yeah, they're behaving correctly, but they're not really proliferating and they might die at any time. Having confirmed that the cells are now capturing the antigen, we then ask the question, are we enhancing antigen processing? So taking the antigen, breaking it down into peptide fragments so that we can present them on the cell surface to activate the immune system. And so to answer that question, we did the exact same assay, but using now another protein called OVA-DQ. So OVA-DQ is an ovalbumin protein that will fluoresce only if it's cleaved. So once you have processing happening, the protein is going to fluoresce. And as you can see in this case, again, the minute you add the Acitox, you see that the signal is actually enhancing in a dose-dependent manner with 50 micromolar or 25 micromolar, giving you the best kind of result. So this was intriguing, interesting, showing that Acitox enhances antigen uptake and antigen processing. So the next question we asked is, we know that the Acuum technology break the endosomes. That's its main potent effect initially. That's the base point we're starting with. So the question we asked then is, in order for the antigen to be cross-presented, to be captured and eventually processed by the proteasome and presented at the surface, it has to make it out of the endosome. So one way to really confirm that this is happening is to use an assay in which you would give a cell a protein that is quite toxic, cytochrome C. So it's a protein that basically will induce cell death. However, if the protein is captured through endocytosis, it's going to be captured in those endosomes, it's going to be degraded, and the cytochrome C will not be able to kill the cell. So as you can see here, if you add, for example, cytochrome C, you don't really get a signal that is different from a cell that is not treated, meaning that the cell is alive. It doesn't really give you any annexin-5 signaling. However, if you mix Acitox with cytochrome C, that's where you see a complete shift, i.e. the cells are becoming annexin-5 positive, and therefore cytochrome C is released in an intact form to the cytosol, and it's then inducing apoptosis. So that's really a clear-cut evidence that we're able to basically drive endosomal break and delivery of the antigen to the cytosol, where it's then acting or behaving correctly. So of course, to really sum up all of these observations into a biological test in vitro, we then conducted an antigen presentation assay where the MSCs are taken. You basically treat them with the AccuTox and the antigen, and then you add T cells that will respond to one specific peptide derived from ovalbumin. And that peptide is synfical. So as a positive control here, you can see that synfical drives the activation of the T cell B3Zs. And of course, if you add ovalbumin on its own, you don't really get that much of an activation, which is normal. MSCs will not cross-present ovalbumin if they're not stimulated to do so. And you can notice here that the minute you start actually adding AccuTox, you start seeing really, and here we're talking about 1 micromolar going up to 50 micromolar. And you can see here that you've got two concentrations that really stimulate potently the cross-presentation ability of these MSCs. So these are what we call the ARM-X cells. So these are cells that are able to capture the antigen, process it, and present it at the cell surface. So this was a great piece of information that we have in vitro. Of course, we digged in a little bit more. We said, okay, well, how are we going to differentiate ourselves from the first generation ARM vaccine? So as you recall, the ARM vaccine was using the A1 molecule. This is the ARM-X vaccine using the AccuTox molecule. But why are we actually generating second generation vaccine? The answer to this is very simple. When we were conducting some manufacturing with the human vaccine cells, we noticed that we needed quite a lot of antigen from the tumor lysates of patients. So we needed something in the range of 0.5 milligram per ml, which it's feasible, but it's a lot of products, a lot of material to request. However, with the AccuTox, what's really impressive is the fact that you can decrease that concentration by 10-fold. In other words, the 0.5 milligram per ml, that's the concentration that usually the A1 works with. If you go beyond that or lower than that, it's not going to work anymore. Whereas with the AccuTox, if you actually decrease the concentration, you can go up to, or you can go down to 0.05 milligram per ml. So 10 times less material is required to still get that antigen cross-presentation happening. So that means you're simplifying really the manufacturing process, you're simplifying the need for tumor lysate material in the clinic, which is already a big leap in the process of development of this vaccine. The interesting thing, however, is that the AccuTox does not work when it's actually used in serum-free conditions or conditions where there is not a lot of protein around. So the reason for that is because AccuTox causes protein aggregation, and this protein aggregation really depends on proteins around it, so they can form this chunk of proteins that are quite big that are captured by the cells, and the cells then will respond to that by driving what we call the unfolded protein response, which is a defense mechanism to really destroy those protein accumulations, not specifically in the cell, so the cell doesn't have to drive itself into apoptosis or cell death. So we're basically tricking down the cell in making it or pushing it to cross-present antigens. So of course, these are interesting in vitro experiments, but the question is, does it work in vivo? So if you transplant melanoma, then you give 3 shots of the vaccines in conjunction with immune checkpoints, are you basically getting some good results? And the answer is yes. As you can see here, the ARM-X combined with the anti-PD-1 generates fantastic control over a fast-growing melanoma tumor compared to the ARM-X alone, so without the anti-PD-1, which still controls or delays tumor growth. But as you can see, if you use regular MSCs with a lysate or lysate and PD-1, you really don't see, I mean, you have some sort of a delay, but not as good as the ARM-X or the ARM-X combined with anti-PD-1. And of course, the survival of this vaccine was actually pretty good using this artificial fast-growing melanoma model. Now, this was done with 0.5 milligram per ml with the same concentration that was used for the A1, so it shows you that it works pretty well at high dose. But then the question we asked is, could we actually lower the dose of the lysate by 10-fold and still get something interesting? And in fact, the answer is yes. You get a very nice delay of tumor growth, a little bit less than the 0.5 milligram, but it's still working very efficiently, and you can still get between 80 and 100% survival over time. So this was Very interesting, because now we have a second generation vaccine that works as good as the A1, but requires 10 times less starting material to really achieve the objective. Of course, nowadays, everybody's talking about, comparing immune responses between males and females, at least in animals. And therefore, we actually conducted these studies in both male and female mice that got the arm vaccine against the melanoma in this case. And as you can see, males in general generate a weaker immune response than females. That's how it is. That's biology. It could be depending on hormonal changes and whatnot. But what you have to keep in mind is that you're always going to have the same kind of response, but it's only the magnitude of the response that will change. So males will always respond weakly to the vaccine compared to females, but the outcome is still the same. Not only that, but we pushed it a little bit further and we started looking at, safety of the vaccine. Are we getting, for example, some side effects? Are we losing weight? And the answer is no. If you look at male versus female mice, the control animals versus the arm X treated animals, you can see that the animals will still increase in weight over time. And in fact, usually we sacrifice these animals when you have 20% loss in weight, which is far from being the case in this context. Not only that, but we started looking a little bit deeper into what was going on in the animals. Are we seeing, for example, inflammation at the injection site? How is the overall activity of the animals? How is the fur looking, the body posture, the weight loss? And really, we were forcing ourselves to see something. We could see a little bit of inflammation at the site of injection, but that's normal. If you just poke a needle in the tissue, you'll see some sort of inflammation, but that inflammation really goes away at a couple of days after, or week 2 and week 3. That was when conducted in male mice. When we looked at female mice, we got a little bit of inflammation, at the site of injection, but eventually it didn't actually show up at the second and third immunization procedure. And that's because, the system was getting used to the vaccine and we're just, priming or boosting the immune response at that level. So the vaccine, in other words, was pretty safe, pretty potent, and no side effects were observed in both male and female mice. So again, the ARM-X manufacturing is ongoing as we speak. We've already interacted with Health Canada. We've identified the clinical team. We're hoping really to be able to start the phase 1 trial by the end of this year, if not the latest early next year, but that's really depending on the funding in order to basically initiate the phase 1 for that program, which again is highly innovative and exciting because it's a universal off-the-shelf vaccine that could be adapted to any cancer. It's not only for melanoma. In fact, we're doing as we speak. We've got fantastic data on pancreatic cancer, which I'll be probably sharing with you in the future. We're doing this on ovarian cancer, and we're going to be also trying colon cancer. So it's really to show you that this vaccine is adapted or could be adapted to any kind of cancer indication, as long as you give us access to tumor lysate. So with this, sorry, I'd like to summarize the findings. So we were able to convert MSCs into antigen-presenting cells using the AccuTox variant. The cells can cross-present efficiently, but they can do that with 10 times less starting material compared to the A1 derivative. The molecule can break the endosomal membrane, so it did not really change the biology of the platform itself. And really, we can use that vaccine for therapeutic vaccination in conjunction to immune checkpoints, which are heavily used in the field of cancer immunotherapy. And of course, the dry runs are being conducted as we speak, to really be able to convince Health Canada and the FDA to give us clearance for a phase 1. So with this, thank you all very much for your time and attention and the invitation, of course, and I'd be more than happy to take questions. And please feel free to communicate with us if you have any questions or inquiries or like to see more data. You can reach our CEO, Mr. Plouffe, or the company directly to get access to these things. Thank you very much.
Well, thank you very much, Doctor. That was a very, very interesting presentation as usual. While attendees are writing questions directly, so please use the Q&A and the chat box to do so. I want to bring you on a path that we didn't touch yet, and that is basically the human side of cancer, the numbers that are behind, the lives that are broken. So first off, Canada just deposited its budget yesterday. Cancer is responsible for over $26 billion in spending yearly
Yeah.
in Canada alone, $40 million population.
Yeah.
How transformative is the work that you're doing in specifically looking just at this, just nothing else, but just at this? What can we expect of in 5 years, 10 years, 15 years when it comes out and it's a product? I have trouble wrapping my head around it.
Yeah. So again, this is something I teach to students at the university. I always tell them, look, our biggest enemy in the world, our number one enemy in the world is cancer, right? You have cancer, you have infectious diseases, but cancer is really, really the number one enemy because this is a disease we have not yet learned to control efficiently. So the reason for these spendings is really because of palliative care, standard of care that is not working, but it's just prolonging the life of these patients. Now, yes, there are some patients that do survive, let's say after surgery, they've got good response rates. There are tumors that are much better handled than others, but there's a lot of cancers that are not or hard to treat so far. So that's why defense is really focusing on pancreatic and ovarian cancer more than melanoma or other stuff. But the thing we're trying to really bring forward here is that, we would like eventually that spending to be done in the correct manner. So in other words, instead of just spending the money and then having, unfortunately, the patient pass away, well, we would like to spend that money so that the patient can survive not 6 months, but 6 years or more. That's the objective of what defense is working on. And in fact, I presented today a second generation ARM-X vaccine, but we've got other projects that are working in the background, which will go way further than that. So the idea now is not really to just treat cancers that are sick. So not to develop only a therapeutic vaccine, but we'll be coming back with something that I think will make a lot of echo worldwide because it's going to be a universal off-the-shelf vaccine that you could use to prophylactically vaccinate people in advance. So you can advance the whole population. You can vaccinate the whole population in advance against 7, 8, 9, 10 cancers. And then you can say to these patients, you know what, come back in 15 years, I'll give you 3 more shots of that prophylactic vaccine. So where we're going with this is, now the FDA and Health Canada want to treat sick patients, and I understand that that's
Yep.
normal. But what we want to do eventually is, I wanna say, like in French, we have a saying, vaut mieux prévenir que guérir, meaning it's better to actually prevent the disease than actually treating the disease because you can actually control it. And I think Gardasil 9 is the perfect example. So that's where we're heading eventually. So these costs are pretty enormous, but they're not really giving us the hoped for outcome. That's what we're trying to really come up with.
Well, this is a very interesting question answer, and on top, it goes directly at my second question that I had as a follow-up, which is basically what in your vaccine is different, for example, from herd immunity, is that basically it's a single case, so every single one must be vaccinated in order to prevent, opposite to what we know, for example, on what happened in COVID and so on, is that it's every single person could then get vaccinated and get protected in that way. So you steal it with your research, which is really good. But that was where I was trying to bring you. So with that, it is 4:30 already. Moutih, thank you very much again for being with us. And again, this time I wanted the Q&A to emphasize that biotech companies are not just there as developing vaccine, but they're there to improve life of people and changing. We all know someone that went through cancer, and if we could give anything for that not to happen, we would. So thank you very much for working into that dream.
Thank you very much. Thank you for the invitation again, and hope to see you soon in a couple of months. Have a great day.
Absolutely. Thank you.
Bye.
Bye-bye.